Bacteria need certain chemicals or elements to survive. Some of these elements are going to be in larger amounts, and then some are going to be as trace elements. Carbon, nitrogen, hydrogen, sulfur, phosphorus, those are all things that bacteria need in large amounts. Phosphorus, or I'm sorry, not phosphorus. Iron and phosphorus are both chemicals that tend to be limiting factors because they're not as common as the other elements. And then we need some elements just in small amounts for different enzymatic functions. Those are the trace elements. So bacteria can have two different, or can gain their carbon from two different sources, either using organic carbon like in the foods, like carbohydrates, stuff like that, or by taking inorganic carbon, carbon dioxide, from the environment, from the air, and fixing it. Some microbes cannot synthesize some of those growth factors. And so they can only grow if we give those growth factors to them. Some microbes can synthesize all growth factors. So E. coli is able to synthesize everything and can grow really effectively in most all environments, so long as we're not putting any inhibitory agents in. Whereas Neisseria is only able to grow if we give it the requirements it needs. Neisseria is considered a fastidious species because it has so many nutritional requirements. Bacteria also gain energy in different ways. So they can gain energy by taking energy from the sun. So that would be a phototroph, or they can get energy from organic materials like the carbohydrates again, and that would be a chemotroph. Humans are chemoheterotrophs because we take organic materials from, or we get our carbon source from organic materials, and we get our energy from organic materials. So here are some questions that I'll let you think about for a second. Remember, heterotrophic organisms are organisms that use organic carbon as their carbon source, and autotrophic organisms use carbon dioxide as their carbon source. They fix it. Phototrophs are organisms that use sunlight for energy, while chemotrophs use organic carbon for energy. So if you are an organism that fixes carbon dioxide from the air and uses organic material for energy, what are you? You are a chemoautotroph. If you are an organism that uses carbon dioxide or uses organic carbon for both carbon source and energy, that's what humans are. You are a chemoheterotroph. Organisms that fix carbon dioxide for carbon source and gain energy from the sun are photoautotrophs. And organisms that use organic carbon for their carbon source but gain energy from the sun are chemoautotrophs. So let's look at the different medias that we have then. If we're going to be growing bacteria, some that are fastidious and some that are very easy to grow, we have different medias for those organisms to grow on. A complex media contains multiple ingredients, but the exact composition is not always known. So you have proteins, but the protein source may not be 100% known. So here's complex media. You have meat extract, but that doesn't tell you what meat. Whereas a chemically defined medium is going to have exact amounts of each type of chemical. And so, there's glucose, or there's monopotassium phosphate, magnesium sulfate. Chemically defined media is what is used typically in research labs so that you are growing the organisms and you know exactly what's in the media, so you know how it will affect the growth of the bacterial species that you're using. Complex media is what we typically use in our lab. We just want our organisms to grow so we can run tests on them. There are a lot of different medias that we can use. Some fun media that we've used in the past that I probably won't use this semester, we've used blood agar. Blood agar is able to grow lots of different species, and it can distinguish different organisms by their ability to break down blood. Chocolate agar is another medium that can grow lots of different organisms, but it's used typically to grow those fastidious species. McConkey agar, I'm going to skip over glucose salts. These are often used in laboratories to grow lots of different organisms, like E. coli. McConkey agar is a complex medium that is selective, so it only grows certain types of organisms, specifically gram-negative organisms, and then it can differentiate gram-negatives on their basis of fermenting lactose. Nutrient agar, that's what we've used in our lab already, which you guys got to do your environmental plate on. This grows multiple organisms. It's not selective or differential. So general purpose medium, these are the media that grow all different types of organisms. Whereas enriched media, they have been enriched by something. So they're a general media that has gained something like blood or a certain nutrient. Some of the media that we'll use in our lab is known as selective media. Selective media inhibits the growth of some types of organisms. So only certain types of organisms will grow. If you look here, you see Serratia marcescens over here, and you see Staphylococcus aureus over here. Staphylococcus aureus are gram-positive organisms. Serratia marcescens are gram-negatives. And if you look on the plate, you can see there's growth right here, but there's no growth over here. This is a selective media that only grows gram-positive bacteria, and it's called PEA plate or phenyl ethyl alcohol agar plate. So PEA is a selective media that grows gram-positives, and it inhibits gram-negative bacteria. The phenyl ethyl alcohol inhibits the growth of gram-negative species. This not only is selective for gram-positives, but it is selective for gram-positive coccus species. Streptococcus, Staphylococcus, and even Micrococcus grow most effectively on this agar. Mannitol salt agar, which you see here, is considered a selective and a differential medium. So a differential medium differentiates among organisms that can grow. So if you grow, then it will differentiate on the basis of utilizing the carbohydrate within the medium. So here's an example of a differential medium. You can see two different types of growth on here. This is blood agar. Blood agar is going to grow, and depending on how hemolysis occurs, how we break down the blood, will determine if you're a pathogenic species or a non-pathogenic species. Beta hemolysis is the pathogenic species, whereas alpha hemolysis is the non-pathogenic species. selective and differential media, they select for specific organisms to grow and then differentiate amongst those organisms that do grow. So this is a mannitol salt agar plate, which you saw a little earlier also. Mannitol salt agar allows for the growth of bacteria that grow in high salt concentrations. So these are the halotolerant species. Staphylococcal species and Micrococcus can grow very effectively in mannitol salt agar. The pH indicator is phenol red, and when the pH drops, the agar turns yellow. So this occurs when bacteria utilize mannitol for their carbohydrate. So this test will select for salt-tolerant bacteria, and then it differentiates the mannitol fermenters from the mannitol non-fermenters. So this is an example of what Staphylococcus aureus looks like on a mannitol salt agar plate. Over here, you see what Staphylococcus epidermidis would look like on the mannitol salt agar plate. I already said that, so I'm not going to go there again. Eosin methylene blue is both selective and differential. It selects for gram-negative bacteria, and it inhibits the growth of gram-positives, and then it differentiates gram-negatives that grow on their basis of fermenting lactose. So here we have a color change in the agar that is indicative of lactose fermentation. Here's another color change, so indicative of lactose fermentation. This one shows heavy lactose fermentation, actually, because it's that green sheen. Whereas over here, we have this clear growth that is showing gram-negative that are lactose negative. Last thing we'll talk about is how we can measure microbial growth. So we can measure microbial growth using direct cell count, where you're counting the number of cells in an environment. So think about taking a sample of sand from a desert, and we want to know how many cells are in that sand. So we can count the number of cells using counting chambers. We can also, but what that does is when we're using counting chambers, we don't always see each individual cell. Or no, that's not true. We don't only see the living cells, we see dead and living. So if we want to know only about the living organisms, then we'd want to do a viable cell count, where we're growing the bacteria, like a plate count, on an agar plate, and only the cells that can grow, because they're living, will grow, producing new colonies. We can use biomass. So this is where you're sending light waves through a broth, and you're determining the turbidity of the solution. And then we can actually use weight, so cellular detection by measuring the weight of the materials that are in a solution. So this kind of shows you the different things I was talking about. I'm going to stop here, and I will see you in class.